Hi, everyone. Welcome to the tech track on precision fermentation for molecular synthesis. I'm David Welch. I'm the CSO and co-founder at Synthesis Capital, and I will be your moderator for this super interesting session. I had the fortune to be at the first GFI conference back in 2018, I think, and while we touched on fermentation there, we certainly didn't have a whole session dedicated to it and diving into the technology. So it's just, I think, an example of how far this particular part of alternative proteins has progressed in such a short space of time. In terms of the agenda for today, if we can move to the next slide, please. We've got a really nice setup. So we'll have two longer talks from Jeroen and James. We may need to switch the order of those just due to some technical difficulties. Then we'll have two flash presentations from Kris at Culture Biosciences and Matt at New Culture. And then we'll have a 30-minute panel discussion that I'll be moderating. So a really, really great format. In terms of, if we can move to the next slide, please. Just in terms of the participation guidelines, you've probably seen these already if you've been in other sessions, but just to kind of remind you, please be courteous when you're in the chat, respect the ideas of others. Please participate. We will have opportunities for Q&A during this 1.5 hour session. So please ask as many questions as you would like, and we'll try our best to get to them. And I think with that, Renee, unless Jeroen has arrived in the last couple of minutes, we might move on to ask James to present.
Yeah, I think we'll go ahead with that plan. Thank you.
All right. So we'll bring up James's slides. Thank you, James, for your flexibility. And maybe while we're doing that, I can introduce James. James is a program manager at the Advanced Biofuels and Bioproducts Process Development Unit, which is part of the Lawrence Berkeley National Laboratory. James has a Ph.D. in biochemistry and molecular biology, and he's done an incredible amount of work in his career in the biofuels area, but more broadly in just supporting advanced biomanufacturing. And I'm sure he will tell us all about what he's done and what he's doing now. So James, with that, I'll pass it over to you.
Am I live?
Yes, we can hear you.
Okay, great. Okay, well, first of all, I'd like to very much thank the GFI for this opportunity to speak. And so a big thank you to Liz Specht, as well as to you, David, for providing me with this opportunity to interact with this tech track today. It's a great chance to present what the ABPDU is all about and some of the food industry work that the group has been involved in. So the Advanced Biofuels and Bioproduct Process Development Unit, or ABPDU for short, and yes, there will be a quiz on this at the end, is a bioprocess scale-up facility located in Emeryville, California. And for the uninitiated, Emeryville is a small town or a small city sort of wedged between Berkeley, California and Oakland. And so by virtue of its location, it's really at the one of ostensibly the hubs of synthetic biology and this very, very fast growing industry. The ABPDU, as David mentioned, is part of Lawrence Berkeley National Laboratory, the main campus of which is actually perched in the hills above UC Berkeley and home to quite an amazing array of talent and world-class science. Of course, a number of Nobel laureates, not the least of whom is Jennifer Doudna herself. So big shout out to all the great work that takes place at UC Berkeley and the Berkeley Lab. So at the risk of coming across a bit salesy, I'll just say that I'd like to go over what the ABPDU offers in terms of its capabilities as a sort of this general purpose pilot plant. And then I'll delve into a few examples of our food collaborations and what having such a facility means for biomanufacturing in terms of dollars and cents, and then introduce you to the team and touch very briefly on the workforce development work that the ABPDU is engaged in as well. So if you could advance the next slide. Okay, so the ABPDU is funded by the Department of Energy's Bioenergy Technologies Office to support industry and academic partners and to scale up technologies and develop prototype level materials. What you're looking at here is part of our 15,000 square foot facility. This is sort of a fisheye view of our fermentation suite, which contains reactors from the 250 mil scale up to the 300 liter scale. So those very early stages of the kind of valley of death of startup and scale up. And so we access over a dozen different kinds of recovery options to sort of bolt onto the work that we do in fermentation, and offer a variety of different kinds of reactors for chemical catalysis and extraction. And I'll get into that a little bit more as we go along. So biomanufacturing is kind of an environment that has two sides that are sort of arguably disparate. Oh, if you could advance the slide, Renee, please. Thanks. So the ABPDU kind of exists at that bridge between what happens at the very small scale that is necessary for high throughput testing and assessment of new technologies, and what happens at those intermediate scales. And those two scales do often behave very, very differently by virtue of the fact that the way liquids interact with the walls of their vessel is entirely different when one is looking at a 250 microliter well versus a 250 milliliter vessel or a 300 liter vessel. And so understanding how to bridge that gap, how to deal with real world feedstocks and minimal media, and how to switch from batch to fed batch to continuous modes are all just some of the things that happen to really drive the complexity and lack of predictability from a very small scale to those intermediate scales. And that's what ABPDU is really all about. Next slide. So we like to say that we just meet our collaborators where they are. If a new startup, for instance, is looking for support to transform shake flask technology, we have multiple bench scale options to kind of accommodate that. And we have worked with many a company who really come to us with an idea, something that looks reasonably promising at the shake flask, and we take them all the way up to the 300 liter. If a company or a group is looking for something more transactional, something where they are looking, say, for a third-party validation at the 300 liter campaign, we're certainly able to offer that as well. So I'll give a quick shout out that we are also developing gas fermentation capabilities now, which hold promise for allowing us to develop products out of gaseous feedstocks as well as cheap renewable electrons. So we're very excited about some of these new technologies that are now coming on board. Okay, next slide, please. And as I'd alluded to, the fermentation side of the house is one part, but we're also really involved in feedstock deconstruction research and using those types of vessels, which are capable of achieving pressure and temperature and accommodating aggressive chemistries, understanding how to leverage those types of equipment for the food space as well. And we've used, for instance, our decanting centrifuge on a number of occasions for supporting our food collaborators, not to mention our other vessels, which allow for interesting and innovative types of extraction procedures. So, so that has been very fruitful for our food collaborations as well. Next slide. Okay, so to fully support collaborators' conversion operations projects, it's so important to really understand what happened.
Sorry, it looks like we just lost James. Please hold on while we figure out our technical problems. Thanks, everyone. Appreciate it.
All right. So yeah, I'll just touch on this very briefly. I do want to spend more time kind of getting into the stories of our collaborations with food. And so really just driving home the point that downstream processing is kind of a mess. There's lots of different things that take place in the DSP space, and each product has its own needs and requirements, and attempting to thread the cost needle is very, very important, and DSP often has an important role to play in that. So we work with our group, with our collaborators on, be it solid liquid separation or various evaporation and drying techniques and all points in between. So if we don't have a piece of equipment on site, we will rent one, and we've done that on any number of occasions for our collaborators. Okay, next slide. And again, I'll touch on this very briefly. Analytical capabilities, we have what most folks would say are the vanilla flavored type of capabilities within the ABPDU, be it various types of separation science, chromatography and the like, as well as some rheological capabilities. And so we bring all that to bear. But maybe the bigger story is the fact that we are part of the Berkeley Lab, and that gains us access to groups like the Molecular Foundry, who are just fantastic in terms of their ability to do a variety of different kinds of electron microscopy and many other types of techniques like that, as well as the Advanced Light Source for X-ray crystallography. And we've leveraged these types of kind of cross-lab capabilities on any number of occasions. So that gives us a lot of firepower beyond just what's in-house. Next slide. Okay, so I want to just touch on a little bit just a few of the collaborations that we have had in the food space. And for this, I'll draw your attention to Perfect Day for milk proteins that we have helped produce, as well as Geltor for collagen, and Joywell for protein sweeteners, Digestiva for digestive enzymes, and Sugarlogix for breast milk sugars, as well as Clara Foods for animal proteins. And so, it's a predictable punchline, but what do all of these have in common? And I'll just say next slide. Next slide, Renee. Oh, I'm sorry. Can you back up? I was looking at the wrong thing. Yeah, this is challenging to do, sharing the deck like this. But okay, so yeah, they're all vegan products by virtue of the fact that they are they have successfully taken kind of the livestock out of the supply chain, as it were, and centered the fermentation tank around the supply chain instead. And so all of these products are amazing in that regard. And so why on earth would the U.S. Department of Energy be interested in this? Well, there's a few reasons, and I'll draw your attention to the first one, and I'll say next slide. Yeah, so the first one is really, especially with the advent of the Biden administration, the real emphasis on decarbonizing the economy. There are any number of reasons why the industry has been moving in this direction, and there are many smart and expert people in the room who can address the other types of issues that are driving the success of this still nascent industry. But there is definitely a real resonance for that decarbonizing piece. Now, beyond that, and perhaps more broadly, I would also argue, and from what I have gathered in all the discussions that I've had with folks at USDOE, as well as folks at Berkeley Lab, is that, overall, the bioeconomy is evolving, and whatever the product is, be it a drop-in fuel or a food product or a commodity chemical or what have you, it's all about evolving the bioeconomy kind of writ large. And so there is a good insight on the part of the U.S. Department of Energy to support the biomanufacturing space in a broad way. And I guess I'll also just mention, and it certainly goes without saying to this audience, that the availability of brand new products that never would have been available otherwise are just such an exciting sort of side of the equation. And so what I want to do now is also bring to your attention with the next slide the fact that ABPDU has a number of case studies that we offer on the website. And so abpdu.lbl.gov is a place where we use as a sort of a clearinghouse for a variety of different kinds of information, not the least of which is these case studies with our collaborators who are willing to sort of open up and share some information further on our website. So I'd like to draw your attention to a couple of these, and then we can jump back into talking more broadly about the ABPDU. And let's see, how am I doing for time? It looks like pretty well, but by all means, Renee, please grab the giant hook from stage left and just let me know when I need to wrap up. So if you go to the next slide, Sugarlogix is one example of the kind of work that we've been doing, and here they're all about developing human milk oligosaccharides by way of fermentation and downstream processing, and these are sugars that are naturally found in human breast milk and have been shown to be incredibly important for healthy immune systems and neurological development in infants. And so a real need is there for this type of product in the market. So Josh Cohen, fermentation scientist at Sugarlogix, was quoted as having said that having a resource like the ABPDU in the Bay Area is extremely useful, and ultimately we were able to have a flexible collaborative relationship. That part in particular was really helpful for generating these important results for our projects. And I guess that really drives the point home for me, in that the ABPDU is still at its core kind of a research institution. We are not kind of strictly speaking a GMO. I'm sorry, a CMO, and that is a contract manufacturer. We don't participate, nor do we want to ever compete with those types of entities, and in fact, we collaborate and communicate frequently with them. But what we do is offer a bridge from that flask scale, those early stages, and provide a way of de-risking as a group moves into a contract manufacturing toller type of environment. So if you can advance to the next slide. So Digestiva is another really interesting case study, and here we make the point that good digestion is a key factor in overall health, and in particular, protein digestion combats muscle loss and improves nutrition and boosts immunity. And so the scientists at Digestiva have developed proprietary enzymes that improve protein digestibility. And this is a big issue, particularly in the developing world, as well as folks with various types of conditions and immunocompromised conditions and so forth. So this has a real resonance for specific types of groups, as well as individuals and communities that don't have access to good, easily digested nutrition. So a really valuable and important aspect to the food story overall. And so ABPDU, this is a great example of one where we did take a shake flask technology all the way to the 300-liter scale, and within a couple of months, we're able to generate a kilogram of product for the Collaborator. And so Joel Cherry, who is the CEO of Digestiva and formerly part of the leadership at Amyris, was quoted as having access to large-scale equipment for fermentation and recovery in combination with small-scale fermentation equipment is not that common. And again, that speaks to that bridge. And so I saw so Joel says, I saw a huge advantage in being able to do proof of concept and flesh out experiments before going to those larger scales. Okay, so why don't we go to the next slide? So the ABPDU has worked with over 65 companies now. This slide is slightly dated at this point, and that crosses from the biofuels and biomass space, sort of where ABPDU got its start, and then has moved into materials and commodity chemicals, and perhaps most recently into the food, health, and ag space. But even there, we're really enjoying a tremendous level of collaboration with companies in that space, and it really runs the gamut from huge companies like Fater, which is essentially the European arm for some of the larger producers of paper products, as well as sort of these recent companies that have come up like Ginkgo Bioworks, but tiny companies as well, like Checkerspot and Perfect Day and others who are really kind of on their way now as a direct function of working with the ABPDU. Okay, so next slide. So production is a huge part of why groups come to the ABPDU, just to be able to have materials that they can sort of offer as proof of principle to prospective offtake partners. And we've worked with a number of food companies in this way: Geltor, Perfect Day, Ripple, and others. So we're excited about that, and it's great that we are able to offer that sort of production capability before groups invest in their own pilot scale capabilities. So next slide.
James, we're unfortunately running a little bit.
Securing private financing, essentially de-risking the whole process of demonstrating the ability for a technology to scale is a big part of what the ABPDU has been able to offer the food sector as well as other important sectors. And so collectively, I'll draw your attention to the white circle. We have been able to really claim sort of a line of sight of access to new capital as a function of work that groups have done specifically with the ABPDU, and to the tune of $960 million and growing. That number is actually a little bit dated now as well. So that's really kind of an exciting and pointed aspect of what the ABPDU is really bringing to the food sector as well as other biomanufacturing sectors. Next slide.
James, I don't know if you can hear me yet.
Can you hear us, James?
Yes.
Okay, great. Yeah, unfortunately, we need to transition to the next presentation. So.
Oh, am I out of time? Okay, I'm sorry.
Yes.
Well, great. Well, thank you so much. I appreciate your time, and I look forward to receiving any questions by whatever means the audience can reach out, and happy to have any conversations that folks are willing to have. Thanks.
Thanks so much, James. And there are a couple of questions in the chat that we didn't have time to get to, but maybe you can answer them in the chat if you wouldn't mind. And I'd like to now welcome Jeroen to the stage. Jeroen is a senior scientist and business development manager at Wageningen University. He also holds a professor chair in industrial molecular microbiology at the University of Amsterdam. He received his PhD in microbiology from the University of Groningen and spent a number of years in industry before joining Wageningen. So he has a really wealth of experience in fermentation. I'm really looking forward to his presentation. Over to you, Jeroen.
Okay, good morning, good afternoon, good evening for people from different parts of the world. Thank you, David, for this nice introduction and for inviting me to be part of this exciting session. So what I would like to do in this 20 minutes is to go over some of the approaches that we are following in the Netherlands, in Wageningen research to produce biomass, protein, and also some other biomass-related components using the process of fermentation. So next slide, please. So this is sort of a schematic view of how we approach fermentation for production of beneficial components and ingredients. So we're talking here about biomass. In some cases, we're talking about pure sugar. In some cases, we're actually even talking about waste gas like syngas. And we use selected microorganisms, here represented in a yellow oval. Basically, these organisms convert these substrates in organic acids like lactic acid or in alcohols like what a lot of yeast do. But there's also all kind of side products which are actually very valuable that are being produced like vitamins, but like preservatives. And that's what the original process of fermentation, food fermentation, has been discovered for. And also now things like flavors and fragrances and sweeteners and pigments. And actually in this topic, I will basically focus on only the 2 points that are related to biomass. This is protein and oils and fatty acids. So next slide, please. So let's say basically all our examples of fermentation and all of the benefits and properties that are produced as a result of fermentation are based on the type of knowledge that we know from food fermentations all over the world. And there's this scheme of fermented foods where all these different fermented foods that have been studied and have been used and eaten for ages in different parts of the world. And all this information that is coming from that and the organisms that are being used and the typical products and flavors and other components that are being formed are the inspiration basically for all the directed and precision fermentations that we're talking about today. So next slide, please. So the approaches that we use here in the Netherlands in Wageningen are shown in this slide, is that we use fermentations from the microliter, the 100 microliter scale in the microtiter plate, to the Erlenmeyer scale, and then the more controlled fermenter scale with the 1 and 2 liter fermenters. And then we have the ability to scale up to 50, 100, 1,000 liter scale all at our facilities, and to basically test for scalability of the process. And the type of applications that we are looking at is conversion of agri-food waste streams by fermentation. We are looking at different kind of properties and products that are being made by fermentation. And we do that for both food and non-food applications. Next slide, please. So the first small story that I would like to tell you is about the possibility to produce oils as a result of fermentation. And what we do in this approach is use specifically yeast and also some fungi that are known to accumulate large amounts of oils intracellularly. We know of some organisms that have almost 70% of their total biomass is composed of oils. And these oils are actually very similar to some of the vegetable oils that we use daily in all our food products, like sunflower oil and palm oil. And we are basically exploring the possibility of using these oils from microbial source as a sustainable alternative. Next slide, please. So basically, the organisms that we work with most are the two yeasts, the Cryptococcus curvatus, which in our hands is the highest producer of oils, but it's not a food grade microorganism. And then we have Yarovia lipolytica, which is actually food grade, which produces slightly less, but is very interesting for food application. And we also are studying various microalgae, which are specifically known for production of oils containing large amount of polyunsaturated fatty acids. So what we do is basically explore the possibility of growth of these organisms on various waste streams and get maximal production of these oils. And we also look at how fermentation conditions and also the different strains that we use can affect the total amount of oils that are being produced, but also the specific composition of the oils, the length of the chain length of the fatty acids that are part of the oil, and also the saturation or unsaturated level of these fatty acids. And next slide, please. Well, if you talk about the application of these oils, I was talking about replacing the vegetable oils that we use in most of the consumer products that we know from the shop, like various food products, of course, but also hygiene and cosmetic products, and there are many more of these applications. And we also know that the fatty acids that are derived from some of these oils sometimes have very specific antifungal activity. And one of the examples where we have worked with is that we have made mayonnaise, which is actually based on only microbial biomass, so both protein and oil deriving from microorganisms. And we also know that, of course, products like meat and dairy products, and when we look at alternatives, we also need alternative oils and fats, and that can also be derived from these microbial sources. And then other possible applications are also surfactants and emulsifiers. We can convert some of these fatty acids into fatty alcohols with very interesting application, for instance, in cosmetics. And we also know that there are plastics or PHA that are based on some of these fatty acids that are produced, hydroxy fatty acids. Next slide, please. Well, the other applications, and that's actually the major part of my story, is about the protein that can be produced as a result of fermentation. Next slide, please. And then in this case, we are inspired, of course, by some of the famous products that are already consumed for shorter and longer times, like corn, which is biomass from the fungus Fusarium. And next slide is an example from the Southeast Asia, the natto product, which is actually containing a lot of Bacillus subtilis biomass and protein. And these examples are an inspiration for some of the products that we're looking at currently and some of the processes that we are developing. Next slide, please. So the approaches that we are following with respect to microbial protein is that we are exploring existing microbial biomass, and that is biomass that is being used for current industrial processes like bioethanol, where huge amounts of yeast are being generated and in many cases are underutilized or not utilized at all. We have large amounts of protein being microorganisms being used for production of organic acids like citric acid, lactic acid, and acetic acid, where we get biomass from the fungus or from a bacterium, bacillus, or from other bacterium, gluconobacter. Also, there huge amounts of biomass are being formed, containing more than 50% or more of a protein, which is underutilized. Another approach that we are following is to look at novel, especially solid-state fermentations, where some of the substrates that we use, like lupine, chickpea, and clover, and various cereals, where we use the organism that is known for tempeh, which is a solid-state fermentation based on soy. We use other substrates in this case, and then also look at how much protein is being produced and what is the type of protein that is being produced. And then we also explore the possibility of producing protein on the basis of residual streams coming from the agri-food industry. And what we do is look at the protein that is being produced, on the composition and the digestibility, and we also look at how different animal models like the zebrafish respond to this protein with respect to motility and the microbiota and the intestinal integrity, to get an idea of how good is the protein compared with the current protein that are used, for instance, in feed, but also in food applications. Next slide, please. So if we talk about the amounts of this protein, the examples that I just gave you here, these are all rough estimations, but the amounts that are being produced as a result of these of microbial protein, are tremendous. And if we would just use only a small part of it, it could also already solve maybe a large part of the protein limitation that we have for many applications and for many countries in the world. Next slide, please. So if you talk about the example of the novel solid-state fermentation, here's a picture of tempeh, which is the organism Rhizopus oligosporus is used for that. And there also the original material is replaced for large part by the fungus, which is the Rhizopus. So the biomass that you find in there is mostly microbial biomass, and there's lots of protein in there from microbial source. Next slide, please. One of the things that we have studied together with my colleagues from the university, Jasper Swinkels and Eddie Smith, we have looked at new substrate, novel substrates, different staple cereals, and I'm not allowed at this moment to share with you which cereals we had used and also which fungus we have used, but we have tested different fungi which are able to perform this solid-state fermentation in different staple cereals, and then we look at what happens to the overall composition. Next slide, please. One of the things that we focused on is especially the amino acid score, where we look at the availability of essential amino acids, and we compare that with the original material. Next slide, please. And here, this is just a short overview of these kind of studies, where we see that various branch chain amino acids and lysine and histidine, that you actually get a higher ratio of these amino acids in the overall product as a result of fermentation. So it means that the overall protein that is offered by this process is different and is sometimes also beneficially better than the original product. And so there is actually a benefit to the production of this microbial protein. Next slide, please. So the type of organisms that we use for microbial biomass based on residual streams are organisms like the Pichia pastoris, the original single-cell microorganism, the yeast, which is actually also a very efficient producer of recombinant proteins. We use Bacillus subtilis, known, of course, and inspired by the food example, natto. It has much faster growth than pigia and sometimes higher yields. And it also can produce some proteins, can excrete some protein very efficiently. And we have this Yarovia lipolytica, which I already presented before as a producer of oils. But this organism also produces quite a lot of protein. And also this protein can be very interesting in some applications. We also have some bacteria, other bacteria, also anaerobic bacteria that are not only interested because they're producing proteins, but they produce also other compounds like vitamins and pigments and flavors. And then finally, one of the organisms that we focus on is Moniella, which is a yeast that produces lots of protein, but it also produces the very interesting low-calorie sweetener erythritol. Next slide, please. And the type of waste streams that we test, and it depends, of course, on the customers that come to us, are things like municipal solid waste, but also all kind of waste streams from the sugar refinery industry. We have many examples here in the Netherlands also of crops that are produced by vegetable growers, and where 25% of that is refused at the auction because they don't look nice or whatever, and then they need to deal with that. And many of these growers want to get rid of these crops as quickly as possible because they cannot store it, and they start deteriorating and spoiling at the location. So they actually pay sometimes to get rid of their refused crops as soon as possible. So they are very interested in getting more value out of these crops, and they are very ideal substrates for fermentation processes. So other examples here are things like seaweed that covers some beaches, and spent barley and potato peels. And there are a number of other examples here which we have all tested as a source for microbial fermentation. Next slide, please. So one of the issues that you deal with when you have some of these substrates and you do a fermentation is an extensive amount of foaming that you get. And here's an example where basically the foam starts to basically spoil your whole process because you don't have the ability to control your fermentation anymore. And this is an example of a Moniella reactor after a couple of days growing in such a substrate, and you need to deal with that. So this is an example of issues that you can face when you are in this area of microbial protein production. Next slide, please. So another issue is when you have these substrates which are heterogeneous, they contain lots of insolubles. It's very difficult to analyze growth of your microorganisms. You cannot follow optical density. Cell counts are sometimes a challenge. It's very difficult to separate microbial dry weight from all the other dry weight which is already in your substrate. So, and protein determination just gives you an idea of total protein. It doesn't tell you if it's microbial protein. So one of the solutions there, and that is specifically for growth with fungi, is to look at indicator molecules. And one ergosterol is an example of a molecule which is specifically present in biomass of fungi. And in this way, you can enumerate the presence of these fungi in such difficult mixtures. Next slide, please. And this is also the approach that has been chosen in these solid state fermentations that I told you about, where we looked at basically the growth of these different fungi in these different substrates. And then you can see that it increases, and that is basically a representation of more biomass being formed. And there's actually a calibration curve that can tell you exactly how much of the biomass is being formed. And there's no very good other way of determining that. And we actually need that also, these kind of indicator molecules for other types of biomass from yeast or from bacterial origin. Next slide, please. So other things that we do with this type of biomass, and these are examples where we used the yeast biomass from Yerovia, which actually contains both protein and oil, and we look at how it behaves in, for instance, different treatments and different cooking processes. And here you can see that we have used it in 3D printing in some cases successfully. And next slide, please. Next slide, please. And also in cooking, we have looked at how is the baking behavior, and does it look or resemble flour in some cases, and also with some of these biomasses, and also mixtures of biomasses from different sources, you can get examples of cooking behavior which is similar to flour from plant origin. And next slide, please. Next slide, please. So finally, I would like to show you also, and this is something that we realize, is that for many of the business cases based on these conversions of side streams into microbial biomass, it depends very much on the product that you get out of it. And we know that, for instance, if you take single cell protein, for instance, based on Candida, we have here a substrate that, and we have done the whole calculation, the technical economic evaluation, and we've seen that only protein production does not really give a good business case, because actually the cost of your fermentation exceeds the revenue of the product coming out of it. But we also see that for some of the examples where you, for instance, use an organism like Moniella, we can get two products. We can get protein and erythritol. And in that case, if you both can get them out efficiently, you can get a good business case, which actually will give you revenue. And we now realize that for many of these business cases based on fermentation of side streams, you need co-production of at least two products to be successful. Next slide, please. So in summary, we know that fermentation from originally for preservation, but we also know that there are many other benefits coming out of fermentation. You can use it as a source of proteins, oils, vitamins, colors, etc. We also know that it can lead to changes, improvements in protein composition of several agri-food products. And we know, we have shown that you can do actually precision fermentation with selected organisms, also based on different side streams. And you need, in some cases, strain selection and culture adaptation to get the most successful fermentation. And you will only get viable business cases for conversion of side streams into beneficial compounds if you look at more than one product being produced based on these side streams. And that's my story, and thank you for your attention.
Thank you so much, Jeroen. That was fascinating, and a great overview of the fermentation space. So now we're going to kind of, I guess, move from an overview to a couple of flash talks on more applied applications of fermentation. So first up is Kris Tyner, who's Senior Director, Bioprocessing at Culture Biosciences. Kris got her Ph.D. in molecular, cellular, and developmental biology from the University of Colorado at Boulder, and she's going to tell us all about culture biosciences. Kris, over to you.
Great, thank you for the introduction. I'm here today to tell you just a little bit about culture and how we work. So if you can go to the next slide. As we know, and we've touched on this morning, developing products for fermentation can be complex because you're developing both strains and bioprocesses in parallel. And what we find is many companies find that they just don't have the capacity that they need internally to be able to support all of the work that you need to do to successfully scale a process. So this is where culture comes in. What we have done is built a cloud bioreactor lab, which is a lab of 250 mil at the moment, cloud-connected bioreactors. And we allow you here to keep your bioprocess development in-house in that you're designing all of the experiments yourself, and we execute for them. And you can follow along your experiments live with us, and so you're able to really be engaged with your experiments as they're happening. So if you go to the next slide. What we aim to do is operate as an extension of your lab. So we're integrated into your R&D workflow as you're running experiments. So how we work is we transfer your processes to culture. So we work closely with you to transfer your process successfully. And then once we have completed a tech transfer process, what you do is you have ongoing guaranteed capacity where you have a certain number of bioprocess runs per month that you have contracted with us. You design the experiments. We execute those experiments in our cloud lab, which again, you're able to follow along with all of the data live as it's happening. You then analyze your data and receive any samples that we ship back to you, and you can use that information to be able to design your experiments. Next slide, please. We begin our process with a structured technical transfer process that we use to build a robust scale-down model for our clients. So this process takes into account the wide range of customers that we work with. So we work with a number of customers at different stages in development and across different industries. We also encourage our customers to share their scale-up plans as soon as possible so that we can also place bounds on the process accordingly. Customers that have worked with us have scaled to many different processes. So from bench scale, pilot, and even to industrial scale. And so you can see on the right here, I can't share, the actual Tyner data, but this is an example from a client that worked with us and scaled all the way from bench scale through pilot scale and eventually up to 50,000 liters. And here you can see that the wet cell weight trends and the biomass yields track nicely between the scales. And we really want our customers to be able to scale successfully with us. And down the bottom, you can see a couple of different quotes from our customers. One of them, from a company that is in the food space. So one of our customers is Clara Food, and they have scaled successfully with us. If you go to the next slide. So our system is designed to be able to replace or support your internal bioreactor capacity for whatever you need to run. We've worked with customers at various scales. So people that are developing their first bioprocesses from shake flask cultivations, people that are doing early process development. We also work with people further along in development. So maybe to refine those correlations between microtiter plates and bioreactor models. People that are looking to do media development and simplify their components. People that are doing experiments to prepare for scale-up, because there's a lot that can be done at small scale to be able to de-risk the scale-up process. We also work with customers that have already gone to commercial scale and maybe help to improve their processes there or troubleshoot problems as they're happening at scale. So we've worked with customers across the spaces and at various different stages. And so the next slide. You can't really see it very well, but you can see our website down the bottom. You can go there to find more information. And thank you.
Thank you so much, Kris. Super exciting what Culture are doing. And Kris will be on our panel in 5 minutes or so. So please continue to ask questions in the chat and we'll try to get to those during the panel discussion. Now I'd like to bring up Matt Gibson. Matt is the CEO and co-founder of New Culture, where he and his team are using precision fermentation to produce animal-free dairy cheese. Take it away, Matt.
Thank you, David. And thanks, GFI, for having me. My name is Matt, co-founder and CEO of New Culture, as David says. And it's honestly a real privilege to be speaking at the GFI conference. We're huge fans of GFI. and we really admire what they do to push the industry forward. So I'm excited to spend these 5 minutes giving you a glimpse of what we do at New Culture. And if you can go to the next slide, please. Okay, so we make cow cheese without the cow. And what does that mean? It means we're using precision fermentation to create real dairy products without any animals required. And that slide you see right there is the result. On top of this pizza, an animal-free mozzarella that melts, stretches, tastes, browns, feels just like the mozzarella we all love. If you go to the next slide, please. And one of the many drawbacks of plant-based cheese that we hear over and over again, and if you've tried plant-based cheese, I'm sure you're familiar with this, is their lack of functionality. And we all know plant-based cheese doesn't stretch. It doesn't stretch because it doesn't contain dairy proteins, specifically casein proteins, which is the hero ingredient that makes cheese cheesy, essentially. And so what you're seeing here in this video is the power of casein proteins. We're utilizing precision fermentation to make casein proteins that enables us to make a cheese that actually stretches exactly like animal-derived mozzarella, as you can see from this video. And I guess it's so good we're playing it twice. Next slide, please. Cool. What's actually interesting about being in the cheese industry is learning not only how old the science of dairy science is, but also how outdated a lot of the equipment and testing methods can be. So especially for the sort of R&D work that we do, when we at New Culture think about rapid iteration and improvement, we think of small scale and high throughput. But what's small scale to the conventional cheese industry is in the hundreds of grams. Whereas for us and other startups utilizing precision fermentation, small scale can be in grams or even milligrams. So we want to get the most juice out of each gram of material. So last year we figured out how to accurately downscale cheese making to single gram scales. And what that allows us to do is to iterate on many different formulations and processes quickly with custom cheese making devices we had to build for ourselves because there actually wasn't anything else out there that we could use. And furthermore, there's a custom melt rig that you see in this slide where after we make these downscaled cheeses, we want to be able to measure attributes such as melts. And we don't want to do it one sample at a time or quantify melt by eye like they actually still do in the conventional cheese industry. So instead, we built our own custom analytical equipment to be able to compare multiple cheeses at once with computer imagery, which allows us to get a real-time accurate understanding of melt and its characteristics. And we can even take color metrics and many other really key analytical data using our custom-built equipment. If you go to the next slide, please. So at New Culture, we're moving as quickly as possible with a very wide array of R&D efforts, essentially in order to commercialize as quickly as possible. The world, I think we all will agree that the world needs more sustainable food. And there's an outcry from the pizza industry for a functional and tasty animal-free mozzarella cheese that actually works well in their ovens, and it can actually be an asset to have on their menu, not a liability like current plant-based cheese is in the pizza industry. So we heard this countless times at this year's Pizza Expo in Las Vegas, which you see pictured here. And, apart from it being the tastiest industry conference we attended, it was actually really insightful for us. And so from talking to dozens of pizza chefs, there's just an unimaginable amount of demand for what we're making, both from the operator and the eater perspective. So, we're already working towards our regulatory and commercial manufacturing targets with a 2023 commercialization goal. If you go to the next slide, please. So on our way to commercial scale, we've done an extended series of tastings of our animal-free mozzarella. Here's a shot from a tasting we actually did a few weeks ago. And the reactions from our tasters, including industry veterans, has been phenomenal. It's been really great to see. And it's important to note that we're not only optimizing our cheese recipe, but also the end products, right? Which is pizza. And we know that the experience of eating our cheese will be first experienced on pizza. So when we think about product optimization and product showcasing, we want to make sure that the entire pizza experience is flawless, including the dough, the sauce, basil on our signature New Culture Margherita pizza, which we call the new classic. Next slide, please. So ultimately, what makes New Culture unique and what makes our cheese cheesy, as I've explained earlier on, is casein. Delicious animal-free cheese comes from the highest quality animal-free casein, and casein is the hero ingredient of all cheese and what gives cheese those functional, textural, and sensory attributes we love about it. So we're using precision fermentation to make kilograms of casein, which we then use to make our animal-free mozzarella cheese with. And what you can see in this photo here, there's just a small amount of our pure animal-free casein protein made using precision fermentation. And it's really this right here, right, that the entire field of precision fermentation is working towards, at least in the dairy space. And that's what allows us to make such good products. And last slide. We believe precision fermentation is the only scalable way to make a cheese that not only has all the functional and sensory attributes we love, but also to create a cheese that actually has great nutritional value while being lactose-free, cholesterol-free, free of trace hormones and antibiotics. And of course, the reason we are doing all of this is to phase out the extremely unsustainable practice of dairy farming and introduce a process that is orders of magnitude more sustainable in water usage, land usage, greenhouse gas emissions, to really give our planet and the next generations a chance to thrive. So that's me. Thank you for listening, and can't wait for everyone to try our cheese in the not too distant future.
Thank you, Matt. I'm looking forward to trying that cheese. All right. So now we've got a panel session coming up. So I'd like to invite the panelists to join me on stage. Hi, Ling.
Hi, David.
Kris.
Hi.
Jeremy. Welcome. All right. So we're running a little bit behind due to some earlier technical challenges, but I think we'll be just fine. So what I'd like to start with is maybe a quick round robin. So if I could ask you all to introduce yourself briefly and your background and the organization you work for, and then maybe just follow with a question. Precision fermentation has such a broad range of applications. So maybe I could also ask you, so what sort of event or piece of knowledge sticks out in your mind as the point where you decided that, precision fermentation and food tech was the thing you wanted to do with your career? So Ling, I see you first, so maybe we can start with you.
Great. Well, thank you for having us here today. So my name is Ling Li, and I lead food commercialization for C16 Biosciences. And so what C16 is doing is building a platform for producing next generation oils, fats, and lipids so that we all collectively are able to remove the most unsustainable ingredients from the foods and products that we interact with each and every day. And the first ingredient that we're actually tackling is palm oil, which is in everything from your peanut butters to your cookies to your chocolates. And so I'm particularly excited actually to be a part of this year's discussions around alternative proteins, because I think innovations around fats and oils is actually critical for the mass uptake of alternative meats. We've talked a lot about quality, taste, and performance over the past few days, and fats and oils, of course, contribute a lot to how those alternative meats cook and taste. So we sort of, as an industry, need fats and oils that function in the way that we need them to. And we also need them to be sustainable. So we're moving to a world where fats and oils that come from animals or messy supply chains relying on deforestation are just not an option. And so that's why it's great to be here. It's a mission that's very closely aligned, requires a holistic solution, from the proteins to the fats and oils to the colors, flavorings, et cetera. And we need to solve it from all angles. So I'm looking forward to the discussion.
Thanks, Ling. Kris. I know we already heard from you, but maybe another brief intro for those that joined late.
Okay, yeah, I'm Kris Tyner. I'm the Senior Director of Bioprocessing at Culture Biosciences. So I just talked briefly before. I think when I knew that I wanted to be in this industry was really mostly for sustainability reasons, understanding that we can't continue the way that we have been, but specifically, I think from a scientific perspective, why I'm here and really enjoy the work that we do so much is that it's just really exciting to see the diversity of products and organisms that people are working with. So even over the last 10 years, like it's just really exploded. And in my position now and previous positions, I've had the luck, I guess, to be able to interact with many different fermentations and really just seeing the different things that people do and the diversity of molecules that you can make is amazing. And it's amazing that, we can do that with biology and technology.
Thank you. Very much agree. Jeremy, hi, welcome.
Hi, my name is Jeremy Chignell. I'm the relatively new GFI fermentation specialist, and I'm so thrilled to be working with GFI, and I get to meet fantastic scientists like yourself. My background is my Ph.D. is in chemical engineering from Colorado State, and after which I went to work for DuPont in Madison at a manufacturing facility there with probiotics, so lactic acid bacteria. After which I moved to Cargill for 3 years, and I was a fermentation scientist there working on precision fermentation.
Thanks, Jeremy. And last but not least, Inja, hi.
Hi, David. Thank you so much for hosting us today. It's really great to take part in the conference, and I must say we're always impressed by the expertise that the GFI brings together for these sorts of events. As David said, I'm Inja. I'm the co-founder and the chief scientific officer at New Culture, and you heard a bit about New Culture from Matt. Actually, probably quite a lot about the product we're making and the tech we're using. So we're making animal-free and real dairy products, starting with cheese, that are indistinguishable from the conventional products in terms of texture, function, nutrition, nutritional content. While they are actually better products, they're better because they are made sustainably, so they're better for humans, for animals, and for our food systems in general. Before founding New Culture, I did my PhD and postdoc research at the LMB in Cambridge in synthetic biology and protein engineering, and my master's at ETH in Zurich in protein biophysics. And to switch into this field, there were 3 sticking points for me, actually, that kind of altogether culminated in my decision to, together with Matt, start New Culture. First was really learning about, in more detail, how unsustainable, how harmful and unethical dairy farming is. We all might have this image in our minds that there are dairy cows strolling across beautiful rolling hills, but that's actually incredibly far from reality. So just like getting into documentaries and books and understanding how problematic this is was crucial. Second was witnessing explosion of plant-based cheese and plant-based dairy alternatives that in case of cheese were really all subpar, and they would not entice consumers to make a switch to a more sustainable option. I kept trying many different plant-based cheeses and just never getting myself to fully enjoy them. So I knew there was this massive gap. And then thirdly, I realizing just that technology is already in place today for us to use precision fermentation to produce dairy ingredients and actually, outcompete animal-derived dairy, both in cost and functionality, in, I would say, less than a decade. So that's what we're doing exactly at New Culture. We're working to achieve this incredible scientific breakthroughs to make this happen, ahead of schedule as soon as we can, starting with delicious mozzarella that Matt talked to you about.
Thanks, Inja. Super interesting backgrounds with everyone on the panel. I'm going to switch to kind of talk about scale, which I think is one of the big challenges and a topic everyone probably in this session is interested in. Jeremy, we're getting a little bit of feedback on your line, so maybe you can hop on while you're perfect. Thank you. So currently, depending on which report you read and believe, that we have somewhere between 60 to 100 million liters of capacity for fermentation around the world. Although much of that, the majority of it, is already reserved, and for most of that for applications outside the alternative protein sector. So, Jeremy, maybe I can start with you and kind of thinking about the growth that we're all anticipating in precision fermentation for the food tech sector. Where do you think we need to be by 2030 to meet demand, and what do you think that capacity looks like? And maybe just an initial question, maybe you can help define precision fermentation for us, because I'm not sure everyone in the session maybe has the same idea of what we mean by precision fermentation.
Absolutely, yeah. And I neglected to mention that when at Cargill was when I really started getting more exposure to the use of precision fermentation for specialty ingredients. They were doing alternative sweeteners, so I really got a sort of a good dose of the power of this technology, and that's why I decided to come to work for GFI, because I really feel like it has the capacity to contribute in a real way. But of course, there's always a but in a sentence like that, and I do feel like we're sort of constrained. I think everybody knows that there's the sense that from a manufacturing perspective, we are pretty constrained in fermenter space. I just did a back of the envelope sort of a calculation, based on that number that David had provided, and so if we wanted to replace all of the milk, all of the milk proteins that are made across the world, 770 billion liters of milk made in 2013. If we wanted to use the existing capacity, say we magically had access to all of that 60 million liters per year that David mentioned, which we don't, obviously, because it's owned by other companies and it's reserved for lots of other products. But say we did, and we wanted to make all of the milk proteins that we currently make from cows. If I calculated it out using Pichia as just sort of a standard organism and, making a few assumptions, so like titers of 5 to 10 grams per liter, which would be pretty thrilling if you're a Pichia fermentation scientist. So we're making casein and other milk proteins, whey proteins that way. We're 3 orders of magnitude low. So that means we would require a titer of around 5,000 grams per liter in order to be able to make the milk proteins that we currently make with cows. And we're at 5 grams per liter, which is sort of a standard titer that we can achieve using standard methods. So obviously we're it's a big challenge. That's just milk. That's assuming that we had access to all fermentation capacity worldwide. So what would it look like for us to try to address this problem? I think that, GFI has really done some work on this to try to establish, we have in our solutions database, if you take a look on the website, a lot of the new sort of solutions that are coming online are associated with contract manufacturing, sort of establishing relationships with the existing fermentation facilities. And I think that that's certainly what it would take to start out with. But I don't think there's any way around the idea that we will need new facilities. We can retrofit existing biofuel facilities. We can, try to build new ones. But I think that really is going to be necessary with that kind of a deficit. So in addition to expanding capacity, we need to, improve the efficiency of our process. So using sort of, the idea being that we're maximizing titer and we're maximizing productivity so that we decrease our cycle times. So your average sort of Pichia fermentation is about 5 days from, turnaround cycle time. So minimizing that turnaround time, maximizing titer. As far as yield goes, or recovery, we want to maximize that. So finding different separations technologies, innovative separations that can allow us to separate our downstream products well. But I would say that if we had to sacrifice on one thing, we could sacrifice on yield. Yield being the quantitation of product per gram biomass or per gram feedstock input. Because as the consumption of animal-based meat goes down, there will be, more corn sugar released for available for other purposes. So I get the sense like,
Jeremy. Yeah. Thanks so much. I just conscious of the time, and I want to give the other panelists a chance to chime in as well. So maybe Ling, I can switch over to you. You mentioned that C16 is using precision fermentation to produce a sustainable palm oil. So can you talk a little bit about some of the main scale-up challenges you're facing with specifically around oils, and how do you see large-scale manufacturing for fats and oils shaping out, say, in the next decade?
Yeah, thanks, David. So fats and oils, just like proteins, just like milk that Jeremy was talking about, are a high volume product. And so we fundamentally think that in order to make a difference in the industry, and particularly for palm oil, we do need to be producing oils at a very high order of magnitude, on the order of a few million metric tons a year. And that's roughly equivalent to the amount of palm oil produced on land that is newly converted from tropical forests to palm plantations each year, with its associated environmental costs. And so it's a tall order. All to say that large-scale manufacturing and the questions around scale-up are very important to us. And it's something that we've recognized from the beginning, and it's actually shaped a lot of the decisions we've taken, both around our strain as well as the way we do manufacturing. And so from a strain perspective, we needed to make sure that our strain was capable of producing oils at the volumes we needed. And so that was important in selecting a strain that was a natural producer, that it naturally likes to produce high amounts of lipids and was very robust in a variety of industrial conditions. And we also needed to get to scale, both so that we can meet the demand and meet the massive and urgent climate need, but also so that we can get our cost structure down. I think that's true for many of us here. We know that it's much more expensive to produce, be it proteins, oils at smaller scale compared to large scale. And the thing that's interesting about oils is, we mentioned contract manufacturing. As we know, there is, a set amount of contract manufacturing capacity available today, not to say it won't increase, but there are some things that are specific to oils, be it from extraction or refining, that not all of these facilities may have. So that's just an additional consideration because we're doing oils that we need to think about. But on the question of how this is evolving, I've already talked about how I do think that the sustainable production of protein and oils markets are closely intertwined. And so I think that as the widespread adoption of alternative meats overall will drive expansion of scaled manufacturing across the board, be that proteins, be that oils, or other ingredients that are needed here. And I expect sort of as that demand increases, and I think it's a need that folks are feeling across the industry, I'm looking forward to seeing more capacity come online as well.
Thanks, Ling. Fascinating. Inja, maybe I can switch to you. We've already heard from Jeremy about like the massiveness of the dairy industry, and also from Ling about what needs to be done around strain optimization. I imagine you're going to say some similar things, but what are the specific challenges you're facing around specifically dairy proteins, and I guess caseins as well, which seem to be fairly challenging to work with compared to whey. And I guess the second part of my question is, you need to almost slot into an existing, well-established dairy industry. So what do you see that space, that industry evolving into in the next decade or so? Oh, you're on mute, Inja.
Sure. Thank you, David. Yeah, you're absolutely right that if we focus on the hero ingredient for producing the casein and look at it from outside of the bioprocess perspective, so just like perspective of microbes producing this protein, this is actually a pretty different and more challenging protein than egg proteins or whey, any globular structured proteins, because caseins are what we call intrinsically resorbed proteins. They are basically not behaving like traditional proteins. So any knowledge that we have on traditional proteins is not directly applied, and they're more prone to issues like proteolysis or aggregation. And I'm really, really proud of the work we've done at New Culture to actually solve for all of those problems and to be able to produce our casein in actually large amounts and formulate our cheese regularly. So yeah, that's definitely been a lot of tough work and creative work and innovation on the strain side primarily. But today, I think it's maybe more relevant for us to talk about the scaling issues you mentioned. And I definitely want to walk through issues we've been having. One learning was that like every company in our field maybe falls under the same umbrella of precision fermentation, meaning we all use fermentation and then there's associated downstream processes to produce and then recover our high value ingredient. And I feel there's not enough talk about the downstream processes, which are actually super important, more challenging in a way. And again, very unique, like every company has something unique to them, something that makes their process better or different or more successful to make their ingredients. So what we have learned actually is that it's really challenging for even the best CROs and CMOs to offer solutions to like all your needs and kind of for all the companies out there. It's really about finding the right partner for you and your process and within the right timing. And that's like very crucial. As I said, something we're very proud of, but that also presents its own issues is that we have actually been moving faster as a company than the industry and available capacity could accommodate. And so CRO and CMO timelines, especially through a COVID time, have been pretty slowed down. And so we really found it challenging to align our internal work with our scale-up campaigns and kind of make sure that we are actually moving on track in both and that we can allocate our resources and time appropriately. So we found it more challenging than we maybe naively anticipated, to be honest. And then there's also, on top of that, there is that inherent time that, again, I think needs to be more talked about. There's inherent time and effort and runs it takes to gather data about your process and about your final product from the quality point of view at each different scale that actually gives you enough confidence to go into manufacturing. So just navigating through all those processes, I think, can be daunting at times if you're doing it for the first time. Now, talking about future and fitting into dairy that you were mentioning, we will certainly be, New Culture will certainly be manufacturing our animal-free casein and dairy products for a number of years in, say, 2030, along with a lot of other companies in this field, I think. And many companies in our field are following a B2B business model, meaning they're making dairy ingredients that will go directly into existing dairy supply chain, meaning being sold to existing dairy companies and other nutritional companies. We at New Culture are taking a slightly different approach. We're actually making our own product under our own brand. However, still, we have the opportunity to collaborate with existing industry and using them as co-mans, for example, for our cheese, right? And really the beauty of producing this ingredient, in our case, animal-free casein, is that it's bioidentical and it can be swapped in for animal-derived casein in the cheese-making processes. So we can easily plug into all the existing dairy manufacturing processes and facilities. And I think that's what we're all aiming to do. We're aiming to work with existing infrastructure and plug into it.
Thanks, Inja. Kris, I'm going to switch over to you, but Inja, just to mention, there's a question for you in the chat, which we may not have time to get to because we're running a little bit late. So maybe you can answer it while we're chatting. But Kris, companies don't just jump into large-scale manufacturing. There's a path they need to follow to be able to do this, efficiently and reproducibly. And we heard in your presentation how your culture, you're improving this, but maybe you can talk about some of the obstacles in general that companies are facing as they come to you. But there was one point that Inja mentioned that I kind of wanted to touch on and maybe have you expand on is data and how important it is to have enough data to make good decisions on what you do next, whether that's bioprocess optimization or strain development. And so I'd like to ask that question to you and then maybe leave you with the closing question as we close this session of what do you think the future of precision fermentation looks like once we solve these challenges with scale?
Okay, thanks. That's a lot there. So, I think a lot of people face challenges with scale up, and I think Inja touched on it with, one of the things is collecting data. You need to be able to generate and collect enough data around your process to be able to scale it effectively. And I think a lot of people have really challenging timelines that they're working on when they're developing processes. Some of the larger, scales, the capacity is small, the timelines are difficult to get in and quite restrictive. So I think in terms of, helping people being successful, we can do work there in terms of just being prepared, right? Understand what data you need to generate to potentially be more successful and anticipate risks when you're scaling up. So for example, you can simulate things like oscillations in substrate or oxygen or things like that at small scale. And so you can have a better understanding of perhaps the robustness of your different strains within a fermentation. And so you can do a lot of testing at small scale that can tell you about some of the weaknesses of your organism and process as you're moving to larger scales. Of course, it won't be perfect, but it's better than kind of going in blindly and just hoping that it's going to scale effectively. Another thing here is also think about your process and what it's going to look like and the constraints it will have when you're scaling up. So for example, a lot of the smaller scale reactors can achieve oxygen transfer rates that are higher than you may see at scale. So just think about the bounds of your process as you're developing it and make sure that you're working within the bounds that would be relevant at industrial scale to make sure that you're, developing something that will scale in terms of those limitations. So I think those are some things that can help people scale. It's inherently complex. Biology is hard. You're looking at an interplay with a strain and a fermentation process when you're developing a product. And it really requires a lot of testing to be able to get to the rate titers yields that you're going to need to be industrially relevant. So I think we've made a lot of advances in terms of synthetic biology where we can produce strains that are producing, really pointed products that we're interested in. And we've also made a lot of progress in terms of how we can test things in fermenters. So I think that leads into, the final piece, like where are we going to end up, in precision fermentation for production of food products. I think the future is bright for the industry. We've made a lot of progress on all fronts, and hopefully we're only going to continue. And, as long as the products, as Inja touched on as well, are, comparable in quality to the animal-based counterparts, I think that people will be excited to try them as well.
I couldn't agree more. What a great way to end this panel session. I see the other panelists nodding as well, so I think everyone agrees. Kris, I wanted to thank all of you, all the panelists and the previous speakers for being part of this fantastic session, and also the audience as well. And thanks for your patience with some of the technical challenges we had earlier on. Thanks, everyone.
Thanks for having us.
Thank you for having us.